Good morning, students. Today we are going to learn about the neuroglia. I have already told that the nervous tissue is made up of neurons and neuroglia, the main components of the nervous tissue. We are studying the nervous system, and in this system there are two types of cells present in the nervous system, that is nervous tissue. This tissue is a very specialized type of tissue. Neurons and neuroglia consist of this tissue. So we have already studied about the neurons, types of neurons also. Now coming to the neuroglia, neuroglia are the supporting cells to the neurons. They are nourishing cells of the neurons. The supporting or nourishing cells are called neuroglia. They are present on the neurons. So there are different types of neuroglia which are present in central nervous system and as well as in peripheral nervous system. There are two types of neuroglia. The chart I have given already. This has been studied in the nervous tissue. Already we have studied about this. Even then I am repeating this one because this is a very important part of the nervous system. The nervous system is made up of neurons and neuroglia. So this tissue is very important. Hence I am repeating once again. I am repeating this one once again because they are very important structures of the system. So neuroglia are of two types. The neuroglia present in the central nervous system. This is the first one, the central nervous system, and the neuroglia present in the peripheral nervous system. So in the central nervous system, there are four types of neuroglia, and they are astrocytes, oligodendrocytes, ependymal cells, then microglia. Once again I will repeat: astrocytes, oligodendrocytes, ependymal cells, and microglia. So there are four types of neuroglia present in the central nervous system. Now coming to the first one, astrocyte. This is of two types, protoplasmic astrocyte and fibrous astrocyte or capsular astrocyte, protoplasmic or fibrous. So these are the protoplasmic astrocytes and fibrous astrocyte, two types. Now oligodendrocyte is a small cell which is present on the axon of the neuron. Axon of the neuron, there is a small cell called oligodendrocytes which is hanging on the myelin sheath of the axons of neurons. Now coming to the ependymal cells, they are very simple cells with cilia. Ependymal cells are ciliated cells. Microglia cells are present in between the dendrites of the axons. So these are the microglia cells. This is the microglia cells which have the spine-like structures at two ends. That is the microglia. Now coming to the satellite cell, that is the microglia present in the peripheral nervous system. In the peripheral nervous system, there are two types of microglia present. The first one is the satellite cell, which is present in the ganglion area. In the ganglions, it is present. It is present in the ganglionic region, that is the synapses region. It is present and the cell is round like this and covered by the sheath of cells. Now the Schwann cell is here, which is covering on the axon of the cell with a groove and cytoplasm and nucleus. So these are the types of nucleus. These are the types of neuroglia. There are major two types of neuroglia present. The first one is neuroglia in central nervous system and the neuroglia present in the peripheral nervous system. Neuroglia present in the central nervous system are again four types: astrocytes, oligodendrocytes, ependymal cells, microglia cells. Here four types of cells. Now coming to the astrocytes, these cells are small star-shaped cells with processes and more abundant in central nervous system. Processes are flattened, leaf-like, which surround and separate the neurons. So these are the cells. Two types of cells are present. They are called as oligodendrocytes. The first one is protoplasmic type and the second one is fibrous type. Now the processes end in expansions on blood vessels or brain surface. They are abundantly found in the central nervous system. Astrocytes are of two types. There are two types of astrocytes. The first one is protoplasmic astrocyte. This is protoplasmic astrocyte. The processes are thick and symmetrical. The processes are thick here and symmetrical. It will be like this. Symmetrical. They are symmetrical in structure. The processes are symmetrical and thick. They are found in grey matter of the brain. The grey matter of the brain contains these type of microglia called as protoplasmic astrocytes. Coming to the fibrous astrocyte, the processes are thin and asymmetrical. The processes are thin. These are the processes of the astrocytes. Which are very thin here. They are thin processes. These processes are thin and asymmetrical. They are not symmetrical, they are asymmetrical. They are found in white matter of the brain. The white matter of the brain contains this type of neuroglia called as fibrous astrocytes. The functions of astrocytes. What are the functions of the astrocytes means? The first one, astrocytes support and embrace the neurons and anchor them to their nutrients supply lines. They anchor the axons or the neurons for the nutrition and for the supply lines. Astrocytes support and embrace. They support and embrace. They are covered. They cover the neurons and they anchor them to their nutrient supply lines. The food is supplied by the nutrients are supplied by the lines, and towards the lines they bring these neurons. They also play an important role in making exchanges between capillaries and neurons. They play an important role in the exchange between capillary and neurons. The capillaries and the neurons are connected by these astrocytes. The processes of astrocytes help in cleaning of neurons and capillaries. The processes of astrocytes clean the neurons and capillaries. They also help in guiding in the migration of young neurons. When the young neurons are produced, these neurons are directed by these astrocytes where they have to sit, where they have to be. So the migration of the young neurons is guided by these astrocytes. They control the chemical environment around the neurons. They control the chemical neuron environment which is present by the side of the neurons. So the neurons have the control of astrocytes for chemical environment, and the chemical environment is maintained perfectly around the neurons. Coming to the oligodendrocytes, these are the oligodendrocytes. The second one, oligodendrocytes. These are pear-shaped cells with few processes. Oligo means scanty. Oligo means scanty. So very few processes are present, and they are pear-shaped cells. They are present on myelin sheath of axons. They are present on the myelin sheath here, on the myelin sheath of the axons. They are present in central nervous system. They are also present in a central nervous system. They wrap their processes around the nerve fibers and produce the main myelin sheath and insulation covering on axons. So the insulation covering, that is myelin, is produced by these oligodendrocytes, which wrap around the nerve fibers. The nerve fibers are wrapped or covered by these oligodendrocytes, and they produce the myelin sheath, which is an insulating covering of the axon. Now the function is, they produce myelin sheath in central nervous system. They provide overall structural framework to the neuron. They provide overall structural framework to the neuron. Now coming to the ependymal cells, these are the ependymal cells. The ependymal cells are ciliated, cuboidal or columnar cells. They are ciliated, cuboidal or columnar type of cells. They are present in the lining of ventricles of the brain. They are present in the lining of the ventricles. Ventricles means spaces of the brain. Ventricles of the brain. Ventricles of the brain and central canal of spinal cord, and also in the central canal of the spinal cord. They are also present in the spinal cord. The spinal cord contains this type of neuroglia. They are called as ependymal cells. So these ependymal cells are also found in the spinal cord. The functions of ependymal, they facilitate the circulation of cerebrospinal fluid, CSF. Cerebrospinal fluid, CSF. This is called as cerebrospinal fluid. The cerebrospinal fluid is present in the spinal cord, and the circulation is facilitated by the cilia of the ependymal cells. The ependymal cells, they are present on the lining of the wall, and the ciliary movement of these ependymal cells helps in the circulation of cerebrospinal fluid, CSF. Now coming to the microglia, the last one, the fourth one, microglia. These are smallest cells of neuroglia. These are smallest cells of neuroglia present in between the dendrites. So they are smaller cells of neuroglia. They are oval in structure with short, thorny processes. These are the thorny processes. So there are thorny processes present at the anterior and the posterior side of the microglia, that is the neuroglia. These are the smallest cells and oval in structure with short thorny processes found in central nervous system with mesodermal origin. They origin from mesodermal layer of the fetus. The cell body is flattened the cell body is flattened. They are frequently associated with capillaries. They are capable of migration, proliferation, and phagocytosis. They are capable of migration, move from place to place. Then proliferation means developing or division. They divide to form the new types, new microglia. They have the reproductive capacity. And phagocytosis. Phagocytosis means they engulf the microorganisms or the microbes which are present by the sides. They fight with the microorganisms or the materials which enter inside the nervous system. The outer microorganisms or microbes are engulfed by these type of microglia called this type of neuroglia called microglia. They have the capacity of migration, proliferation, and phagocytosis. Functions: microorganisms and dead neurons are removed by the phagocytic action of these cells. I have already told they are phagocytic in nature, and they remove the dead neurons and the microorganisms which enter inside the nervous system by the phagocytic action of the microglia. They clean the neurons. They also clean the neurons. Now coming to the neuroglia in the peripheral nervous system. In the peripheral nervous system, there are two types of neuroglia. Two types. ಅಲ್ಲಿ ಎರಡು ಟೈಪ್ ನ್ಯೂರೋಗ್ಲಿಯಾ ಇದಾವೆ. Peripheral nervous system, PNS, peripheral nervous system, CNS, central nervous system. There are two types. One is satellite cell, then second one is the Schwann cells. The scientist Schwann has invented these cells, and the cells are named after Schwann. So there are two types, the satellite cells and the Schwann cells. Now coming to the satellite cells, these type of neuroglia cells are present on the external surface of peripheral nervous system neurons. So these type of satellite cells, they are present on the external surface of the neurons of peripheral nervous system. They surround neurons, forming a capsule around it. They surround the neurons, forming a capsule around it in sensory, sympathetic and parasympathetic ganglions. So parasympathetic and sympathetic ganglions I will explain afterwards. What are the sympathetic and parasympathetic ganglions? So these are present and they surround the sympathetic and parasympathetic ganglions. They are highly sensitive to injury, inflammation and chronic pain. They are highly sensitive to injury, inflammation and chronic pain. Functions are: they regulate the external chemical environment of the neurons. They regulate the external chemical environment of the neurons. They are similar in function to astrocytes. They are similar in the function of astrocytes. They work like astrocytes. They play an important role in modulating the peripheral nervous system following injury and inflammation. So during the injury and inflammation, they modulate the peripheral nervous system. They heal. They modulate means they heal the peripheral nervous system. They provide physical support and nutrition to the neurons. They provide physical support as well as they supply the nutrients to the neurons. They absorb excess of neurotransmitters. The neuroreceptors which are transmitted through the neurons to the brain, if they are excess in type, if they are excess, then they will be absorbed by the satellite neuroglia cells. So satellite neuroglias or satellite cells, they absorb the excess neurotransmitters. Coming to the Schwann cells, second one, Schwann cell. The cells are oval, elongated with a prominent nucleus. They are elongated and oval with a prominent nucleus and nucleolus. Prominent nucleus. A groove is found on the cell. There is a groove found on the cell. They are present on unmyelinated axons. The axons which are not covered with myelin sheath are covered with Schwann cells. So they replace the myelin sheath. The unmyelinated axons are covered by this type of cells called as Schwann cells. They cover the unmyelinated axons. The unmyelinated axons invaginate into the cytoplasm of the Schwann cells. The unmyelinated axons invaginate into the cytoplasm of Schwann cells. Now coming to the functions of the Schwann cells, they provide myelin sheath to axons in PNS. In the peripheral nervous system, they provide the myelin sheath to the unmyelinated axons. They are similar in function to oligodendrocytes. They are similar in the function to the oligodendrocytes and microglia cells. Oligodendrocytes and microglia cells, these two types, the functions of these is carried out by the function of the Schwann cells. They are similar to oligodendrocytes and the microglia cells. They also have phagocytic activity. They are phagocytic in nature and remove cellular debris which help in growth of peripheral nervous system. They remove the cellular debris, the cellular remnants, the dead cells or the dead neurons. All these are removed by this neuroglia called as Schwann cells in the peripheral nervous system. In the peripheral nervous system, we see some neurons are dead, some cell debris will be there, and all these are removed by Schwann cells by their phagocytic action, engulfing action. Now, the last one is the functions of neuroglia. It is very important. The functions of neuroglia. The myelin sheath produced by neuroglia cells which wraps around axons speed up electric impulses conduction. So the impulse conduction in the neurons is speed up by the myelin sheath which is produced by neuroglial cells. The neuroglial cells produce the myelin sheath, and this helps in the speeding up the electric impulses conduction in the neurons. It provides nutrients to neurons including oxygen. See, they are very important. Neuroglia are very important. They provide oxygen and nutrients, food materials to the neurons. They nourish the neurons. They destroy pathogens by phagocytic actions. They destroy the pathogens entering inside the nervous system by their phagocytic action. They provide mechanical support structure in which neurons can sit. They provide mechanical support structure. The support structure is provided by this neuroglia in which the neurons can sit, on which the neurons can sit. They are good conductors as myelin sheath works like insulator. They are good conductors. They are very good conductors as the myelin sheath works like insulator. They prevent spread of impulses. They prevent the spread of impulses. They can repair the neurons by a process called gliolysis. The gliosis is the process where they repair the neurons. They help in growth of brain. They help in natural functions, normal functions of the neurons. They help in the growth of brain and they help in the normal functions of the neurons. They act like barrier between brain and blood. They act like the barrier between the brain and the blood. So these neuroglia are called supporting cells for the neurons. So they support the neurons. So thank you very much.